Good morning, everyone. Welcome to another edition of High Rounds. I think we have a lot of joiners today because we have a very exciting topic and speaker. I guess we want to make sure also, Joel, you can share your slides.
Yeah, I can do that.
You okay? Okay. Well, but before I introduced Dr. Joel Wertheim, I just wanted to welcome some of the um, the applicants, the ID fellow applicants who are joining us today. So just wanted to say thanks for joining Laura, Michael, Nuzat, and Sarah. We hope your day is good and hope it gets off to a great start with a cool presentation by Dr. Joel Wertheim, who is an associate professor of medicine in the Division of Infectious Diseases and Global Public Health. He received his PhD in ecology and evolutionary biology in 2009. His research uses viral molecular epidemiology to track pathogen spread and inform public health action, focusing on HIV, SARS-CoV-2, and more recently, Mpox virus. So I will pass it on to you, Joel, and I'll keep an eye on the chat, and hopefully I'll give you a reminder about the end of the time. We're getting closer to the
Oh,
end of time.
all right. Thank you so much. All right. So am I big for everyone, Jill?
We don't see any slides. Is that what you
Oh,
were? I know you do
no.
ugh,
There we go. All right, we're good.
fanciest slides in the whole world. One day he'll give us a tutorial on this.
All
Thanks.
right. Thank you for that nice introduction and good to be back at High Rounds. Today I'm going to talk about the rise and fall of the 2022 Mpox outbreak in New York City. And we're going to it's almost a career retrospective because we're also going to look at this relationship with HIV transmission networks. We've managing to bridge these two fields. So first, I want to acknowledge all of my co-authors. This is a paper that's currently in revision. So if you have feedback for me, now's the time to give it. I especially want to point out my former graduate student and postdoc, Jonathan Pekar, who led this work, as well as our other collaborators, including Jade Wang, who was instrumental to performing this work. She used to be at the New York City Health Department, and now she's a UC San Diego grad student. So thank you, especially to the both of them. I have no conflicts relevant to this talk. And I'm a big metaphor guy, so I'm going to start with this one, which was Muhammad Ali in the Rumble in the Jungle, where he basically did the rope-a-dope and managed sort of a memorable KO. And I want to come back to this slide, so just keep this image in your head throughout the whole talk. And in the end, it's going to make a lot of sense. All right, so we're going to talk today about the Mpox virus, and specifically the Clade 2B version of this virus that spread around Europe, North America, South America, and Asia in 2022 after emerging from West Africa. And the reason someone like me is interested in a DNA virus is because this one gets hypermutated. So some of you might be familiar with Applebeck from HIV. It's an anti-viral defense mechanism that every cell in your body possesses. And one thing it does, it tries to hypermutate viruses in order to essentially make them non-functional. What this has the effect in Mpox virus is increasing the mutation rate or the substitution rate to a level where we can actually track its spread in real time. It's not quite as fast as an RNA virus like HIV or SARS-CoV-2, but it's fast enough that we can look at its spread between its first jump into humans in 2017 to its eventual spread around the world in 2022. And there was a really excellent paper that explored this global transmission dynamics here out of University of Washington, led by Miguel Paredes. It was published in Cell, and they constructed this phylogenetic tree, which showed the genetic relationship between Mpox virus sampled all around the world, showing repeated introductions into different continents. Here, they divide the world basically continent-wise, North and South America, and then Europe was divided into Southern and Western Europe because, well, that's how Europe does things. But they did draw some very, very important and interesting conclusions here. And the first was that Mpox transmission in North America, directly quoting from them here, began declining before more than 10% of what they categorize as high-risk people, or gay and bisexual men who have sex with men, had vaccine-induced immunity. So basically 90% of the population is still susceptible, and yet the virus begins to decline. And they speculated, although did not state emphatically in their paper, that other factors other than the vaccination campaign may have contributed to this decline. Now, I'd like to point out that their approach considered all of North America as a whole here, US, Mexico, and Canada, and the timing of the epidemics and the peak of these epidemics and the rollout of vaccination campaigns varied dramatically across this area. For example, the San Diego Mpox epidemic was still climbing as the New York City was decreasing. So we decided here to focus on New York City. Now, although most people from New York, and maybe our new division chief will back me up on this, tend to see themselves as the majority of all of North America, as illustrated by this classic cartoon. But in fact, it is just a vibrant metropolis with the largest population of men who have sex with men in the United States. And as a result, had the largest Mpox outbreak in the country for any jurisdiction in 2022. Yeah, Jill's also from New York. With 3,800 known cases and presumably a bunch of other people who were infected but not diagnosed. And you can see the trajectory here if we sort of zoom in on this for a moment. You can see that the epidemic peaks around late July, beginning of August, and then by it starts in late May, early June, and then by December and November, we only have sporadic cases. Okay, now that 3,800 cases is a lot, but it's only 1.5% of estimated number of MSM that live in New York City. So in fact, a lot less than 10% total. So how does the epidemic peak when only 1.5% of all people have been presumably susceptible people have been infected? Well, one option here is vaccination. And you can see here we have it's a 2-dose vaccine where the doses are given about a month or so apart. And you can see the introduction of dose 1 starting in mid to late June, and then really peaking in the beginning of August. These are from the New York City Department of Health. And then by late August, going forward. So that's 154,000 cumulative doses, 100,000 of dose 1, and 550,000 of dose 2. But if we look at this, the sort of introduction of the vaccine over the case count, we can see that the vaccines don't really precede the drop in cases. Yes, they start to ramp up as cases peak, but the timing here isn't highly suggestive of vaccines playing the decisive role here. And this is sort of what we want to investigate. Specifically, why did Mpox peak in New York City at the end of July 2022? Thank you, Jeff, in the chat for anticipating. I think maybe it's my next slide. Okay, so one option is the vaccines did it. So we need to take that very seriously. There was really good vaccine uptake in New York City. Another option, of course, is behavioral change. Maybe people saw the increase in Mpox cases and decided, you know what, I'm going to decrease sexual activity until cases go down, which, of course, I think nobody can deny that that was certainly happening. But the question is, was that enough to change the course of the epidemic? And the third option that I will pose to you is this slide, and I'm going to make it nice and big because I'm contractually obligated to display this figure in every talk I give for the rest of my life. This is the HIV Global Transmission Network, which has some unique features that are important for sexual transmission, specifically its cluster size distribution. So we'll come back to that in a little bit. First, let's just talk about these cases now. New York City, although having the largest single epidemic or outbreak in the epidemic with nearly 4,000 cases, there were more cases in the rest of the United States, shown in dark blue, and also in the rest of the world, primarily in Western and Southern Europe, shown in this gold. And we're just categorizing all of those under global. That includes South America, Eastern Europe, Asia, and Africa. So any case that isn't in the U.S., we're just lumping there in gold for reasons that will become clear later. And it's also important to see that the New York City outbreak peaked before the rest of the United States and before the rest of the world, which is why New York is so interesting here and why it may be the best place to investigate our particular question of why this peak happened when it did. The other great thing about New York City for investigating Mpox is the number of viral genomes that they sequenced. So the Mpox genome is quite big. It is just shy of 200,000 nucleotides long. So this is a very large DNA virus. And New York City sequenced quite a lot of them, over 1,000 of them during this time period, which is more than were sequenced across the rest of the United States. Especially during that particular time period. And you can see at some point even outpacing what was sequenced in the rest of the world. Now, if we consider the number of cases and the proportion of genomes that this represents, New York becomes an even more enticing place to look to answer these questions. Because in the USA, we sequence outside of New York City, we sequence about 4.6% of all cases had their genome sequenced. And globally, about 4.1% of cases had their genome sequenced, which for infectious epidemic, it's actually really good, especially given the difficulty in obtaining these samples early on. But in New York City, they sequenced nearly 20% of cases had a genome sequence. So we have a really nice sample here. Now, I should point out that most of these cases that were sequenced came from the early numbers in when the cases were being diagnosed by the health department, and we had fewer sequences when cases started being diagnosed in labs outside of the health department facilities and diagnosed by Quest or Monogram. All right. So what does someone like me do when they get 1,000 genomes of a new viral outbreak? They throw them into this program called BEAST, which basically reconstructs the evolutionary history of these viruses and their transmission dynamics. And what we can do here is we can build a phylogenetic tree where all of the tips represent a sampled virus, and those branches going back reflect the evolutionary history. And we can see here that we have them colored by, if they're sampled in New York City in red, the rest of the United States in blue, and the rest of the world in gold. And from that, you can see sort of the way the virus moves around the world and the country and in and out of New York City. So we can highlight them in blue there. All right, so what do we do with something like this? Well, we're going to employ a method called phylogeography, which aims to identify the introductions of this virus into New York City. Now, this is a very general audience, so I'm going to take a step back from Mpox right now and just give you an idea of how are we defining what is a cluster of viruses of Mpox in New York City. And we're doing that using a phylogenetic tree. And here we have our the classic example I like to use of great apes. We have chimpanzees, bonobos, humans, represented by a young and beardless Charles Darwin, and gorillas. And what's notable about these four ape species, if you consider Charles Darwin in particular, is that three of them are found in sub-Saharan Africa, and one of them was typically found in Europe. So the question is, where was the ancestor of all of these apes, and how can we show that phylogenetically? So we can start by sort of reconstructing the history, the geographic history of these animals across the tree. So we can go back in time with chimpanzees and bonobos. And as we go deep and up the tree, we find that they share a common ancestor, and that we can then infer that that common ancestor lived in sub-Saharan Africa. We can also follow gorillas back deep into the evolutionary tree and find that they also share a common ancestor with chimps and bonobos, and we can infer that that ancestor was also in sub-Saharan Africa. And then we see here that the ancestor of humans and chimpanzees and bonobos, again, most likely in sub-Saharan Africa, which means along the branch to humans, there's a transition between sub-Saharan Africa and Europe. So we can then infer there was an introduction of apes from Africa into Europe, which is why Charles Darwin was found in England. And now if you just imagine the same thing, but with viruses sampled elsewhere in the world, we're going to be looking for clusters of virus that got introduced into New York City. And from that, once we identify those clusters, we can say, here we have the number of introductions from into New York City, say from the rest of the world, shown in gold in this solid gold line, and then we can have exports from New York City into the rest of the world, and also exports from New York City to the United States, shown in the dashed blue line, and then the solid blue line is the number of imports from the rest of the United States into New York. And you can see that New York got most of its importations from outside of the US, and most of its exportations went to the rest of the United States, which is really kind of exactly what you would expect. And probably didn't need thousands of genomes and a fancy analysis to tell you that's probably how the virus spread. But what we're really interested here is looking at clusters, and those are distinct introductions of the virus into New York City on our phylogenetic tree. And what we can see is that we identify them here. So every time you see the tree go from orange or red to blue and then stay blue, that's a cluster. If it's a single introduction, then it just no evidence of onward transmission, or if it's multiple blue genomes closely related to each other, that's a cluster that then persisted in New York City. And in all, we find about 84 clusters of these viruses in New York City from about 730 genomes that were from New York that were included in our analysis. So what this means is at least 84 distinct introduction events into New York City, about half of which we find evidence for onward transmission. The largest of these clusters comprised 85 people, indicating a long-term sustained transmission of the virus from just a single introduction into New York City. And if we look at the sizes of these clusters, you can see their introductions plotted going back into late April, early May, all the way here through July. Now, these are clusters with at least 2 people. And then you can see that the largest cluster, those colored in red or in dark blue, um, tend to be uh, arrive earlier in the outbreak than later. So the biggest clusters showed up it basically end of April, early May, but really before the start of June, except for one. And those are clusters that we can find at least 11 to 30 people in them. Um, which makes sense because as the epidemic peaked, clusters don't have a chance to be as big. Now, what I think is really important here is the size distribution of these clusters. And you can see that, well, most of our clusters are very small, containing either just a single individual or two individuals. We have what appears to be an excess of clusters that are very large with 10, 20, 30, or as I mentioned, even 85 individuals in them, which is very consistent with what you'd expect looking at clusters of sexually transmitted infection. Now, it had been hypothesized previously based on interview data in the UK that the Mpox virus would be transmitting over a sexual network, which tends to have an excess of sort of densely connected nodes. Some people have a lot of sexual contacts, and some people have an excess of the expected distribution, and more than you'd expect based on the even say Poisson distribution. So what we're seeing here is something that we call a scale-free distribution, or something that could be approximated by a scale-free distribution, which essentially means that we have sort of more highly connected individuals than would be anticipated, and you end up with sort of larger, more densely connected clusters. Now,
Well,
this
there
is
is a
something,
question just
yeah.
to, I don't know if
Oh,
you see
yes,
the question.
I see
Yes.
that. Okay, someone asked,
Do
does
you
that
want to
mean
read the question?
85?
Yeah.
Yeah, does that mean that 85 people were infected by the same person? No, it does not. What it means is that 85 people got infected as a result of a single event and can be connected in a sort of larger transmission cluster. But that could be just a series of, one to one to one transmissions 85 times, or some people getting it from the same person, but it does not mean that 85 people got it from a single individual. Thank you. That's an important clarification to make. All right. So going back now to another STI that has been very well studied, HIV. And with this seminal paper by Andy Lee Brown, published in 2011. Andy Lee Brown was the founder of my group here, UC San Diego, many generations before I arrived. But he published this important paper on HIV, which was the first to look at the size of transmission clusters for HIV and plot out that distribution. And what he found was that HIV had an excess, HIV transmission clusters had an excess of very, very large transmission clusters. Although the majority of these clusters were of size 2, they found many of size, in the 40s, 50s, and even one with 106 individuals in it, showing that sort of excess of very large clusters, which you would see if you have a small subset of individuals who are highly connected in the sexual network. And when he went to fit distributions, find the best fit distribution for those cluster sizes, he found that they were best fit by something called a Waring distribution, and second best fit by something called the Yule distribution. And both of these are these preferential attachment models where people who are highly connected are in fact even more highly connected than you'd expect, as opposed to sort of your classic sort of 80-20 Poisson, where 80% of the people have 20% of the connections. These distributions are even more extreme. And when we looked at this same pattern across all of HIV, shown in this modern art here, we find the same pattern is true, that HIV across the entire world tends to follow this Waring distribution, where we have an excess number of very large clusters, and this is been a very sort of stable depiction shown here in the black line of the HIV transmission network now for over 2 decades. And work done here at the AVRC has shown how this feature of HIV transmission allows for prioritized interventions where if you can find the people or subpopulations that are most likely to be infected with or transmitting HIV and prioritize services to those groups, you can preferably disrupt onward transmission because it's a core group of people in the sexual network that are generating most of the new infections. Okay, so let's focus on HIV in New York City in relation to the Mpox outbreak. So currently we have about 76, almost 77,000 people living with HIV in New York City that have a reported viral genotype that we can use to construct transmission clusters. And we'll do that using a slightly different approach than for Mpox. We're going to use HIV trace, which looks for pairs of individuals with very, very highly similar sequences and connects them into clusters in the same way you would use contact tracing to reconstruct a sexual network by finding pairs of individuals who are linked and then expanding that until you have a cluster of linked individuals. Here we do the same thing, but using the genetic sequence. And from that, we can identify 4,200 clusters of HIV transmission in New York City, going all the way back for people who have a sequence reported since 2005. And in total, out of the 730 people that we had an Mpox genome for, 328 of those people had a concurrent HIV, were also living with HIV and had a reported HIV diagnosis in New York City. So about 43% of people with an Mpox genome also had an HIV infection. Now, what's very notable here is the timing of these HIV infections. So here's the distribution of those cases who have an HIV diagnosis and whether or not they were also in an Mpox cluster. So if they were just sort of a singleton in the Mpox tree, they're in blue. If they're in a cluster of 2 or more individuals, they're in red. And you can see here the year that they were diagnosed with HIV. And you can see the very small number of people here who were diagnosed in 2021, 2022, or 2023. So most of these people who acquired Mpox had been living with HIV for quite some time, in fact, up to decades. And if we look at the relationship here, we can see that if you have an HIV diagnosis and you also received an Mpox genome sequence, you are actually, your odds of being in a cluster, your odds of being in a cluster for Mpox were about 1.58 compared with not being in a cluster. So HIV diagnosis was associated with sort of increased transmission of Mpox after it was introduced into New York City. We found no association with having a reported HIV genotype or actually being within an HIV cluster itself. But we did find an association between being in a growing HIV transmission cluster, that's an HIV transmission cluster that had a case added in the previous year, and being in an Mpox cluster. And both of those are indicative of active sexual networks. But of course, our numbers are quite small here. But notably, of the 32 people who were in growing HIV clusters, 31 of them were also in an Mpox cluster, and only one of them was sort of a singleton in the Mpox transmission network, indicating no evidence of onward transmission in New York City. Now, what's somewhat notable here is that of these 2 networks, we saw that 81 of these clusters we had shared nodes between Mpox and HIV networks. Um, so you can, sorry, go back and see here. Um, we have 81 people who are clustered in both the HIV network and the Mpox network, which means they appear in both networks and are clustered. But this is really important. Zero shared edges between these two networks. That means you're in an HIV cluster and you're connected to people in the HIV network. You're in an Mpox cluster and you're connected to people in the Mpox transmission network. But the people who you're connected to are not the same. We didn't see a single evidence, a single pair suggesting that the same person who transmitted a transmission pair for HIV would be the same transmission pair for Mpox. And the reason for this is the vastly different time scales that these viruses transmitted over. For the population we're looking at here, most of these HIV transmission events are occurring in the 90s and 2000s and the 2010s, whereas Mpox is all happening much, 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 much later. So what most likely has occurred is a rearranging of the sexual network between the time that these two viruses transmit. Now we see, in fact, a very similar pattern when we look at things like hepatitis C and HIV. So viruses that transmit along the same time scale have similar transmission dynamics, but between different individuals as the sexual network evolves and rearranges. Okay. Now for the sort of kind of mathy bit here. We looked at the types of distributions that best fit the Mpox network. And you can see here I have 6 different distributions: binomial, exponential, negative binomial, along with power law, Pareto, and Yules-Simon. Now, the 3 that are highlighted in red, those are those scale-free distributions where you have preferential attachment models. And we can see that the best fitting distributions for the Mpox network, whether by classic AIC or by a method called KL divergence, we can see that the best fitting networks to Mpox are also these scale-free models. And when we just do the same thing for HIV, we can see that again, our best fitting models here ranked by KL divergence are all the scale-free distributions, indicating that both Mpox and HIV are transmitting along this preferential attachment sexual network, which is inevitably going to govern their transmission dynamics. So, just to take a step back here, we then went to characterize the effective reproductive number of this virus, of Mpox in New York City, over the course of the outbreak using a phylogenetic approach, looking in 1 week windows to understand when did RE actually drop below 1 and when did the epidemic, the outbreak sort of peak. And you can see that here, actually, where we infer an RE between 2.5 and 3 for the throughout June, and then in July we start to see a very precipitous decrease in the effective reproductive number, and by mid-July, the virus's reproductive number is well below 1, indicating that the epidemic has peaked and is ending. Now, if we overlay that with the distribution of vaccine doses, we can see that the epidemic peaks long before we have substantial distribution of even the first vaccine dose, which you may not even expect and this is the date of the dose being given, and you would expect you would need 1 or 2 weeks to see any immunological protective effects here. So we can see here the epidemic is peaked and is in precipitous decline before the rollout of vaccine dose 1. Okay, so what do we think actually happened here? To do this, we tried another network simulation approach where we allowed the virus to spread over a simulated scale-free network. and we counted the number of the cumulative number of infections among MSM in the population. And you can see here plotted in the middle windows where we inferred to have that RE inflection point. Now, we're also showing the cumulative infections among the most highly connected individuals. And I just want you to focus here on this red line, which are the 1% most highly connected individuals in the sexual network. And you can see here that by the time 1% of total population of MSM would be infected, the 1% most highly connected individuals, 25% of them have been infected. So the most highly connected individuals in a sexual network are acquiring this virus 25 times more rapidly than the least connected individuals, which would allow you to saturate the most densely connected parts of a sexual network and lead to epidemic decline when only 1 or 1.5% of the total population has been infected. So basically, it's the network. And this is really, really, really important. What about the vaccine? Well, I would argue that the vaccine did not play a role in bringing about a decline in this outbreak. Before people get very upset with me, I want to clarify exactly what I mean by that. If you got vaccinated and then you got Mpox, almost certainly led to improved health outcomes, less serious infection, potentially less likely to transmit the virus. Also, the vaccination that came in August and September could likely have played a role in preventing resurgence of the virus. And also it could have possibly accelerated the decline of the epidemic. That's not a factor that we investigated here. So what were vaccines in the 2022 Mpox outbreak? They were the punch that Muhammad Ali didn't have to throw because his opponent was already going down. Vaccines would have been effective, but the outbreak was already over by the time they were widely distributed. Even in New York City, that had some of the earliest distribution of these vaccines. I told you I was a metaphor guy. All right. Now, what about behavioral changes? I saw in the chat that naturally we're talking about a population that is one of the most responsive to public health messages in the country. So yes, it was almost certainly behavioral change that occurred. What our results here show is that you don't need to invoke behavioral change to get a peak at an epidemic after 1.5% of the population has been infected. So it's not that it wouldn't have slowed the rise and accelerated the decline, but it's not an assumption that's needed in the model to have this virus peak at 1.5%. And unfortunately, we don't have, I haven't seen great data on this in New York City, but I'd be very interested in looking to see how behavioral change could have contributed, and of course, how it relates to resurgence. One other very important factor that we didn't include in our model is a paper that just came out last week out of the Oxford group, Christoph Fraser, again, HIV researchers pivoting to look at new viruses. And they were looking at the difference between symptom onset and delay in presentation on the healthcare system. And what they found was that in the beginning of their epidemic, in the month of May, there was a mean of 22 days between symptom onset and diagnosis. And that by June, that was decreased to 8 days. So just meaning that when we look at when people are getting infected and diagnosed. In those cases, we're talking about anywhere between 1 to 3 weeks, um, uh, predating when they postdating when they likely actually started to be sick, let alone when they first got infected. So they argued that the decline that they saw in the UK was also not due to vaccination and most likely a combination of immunity, which they attributed to pre-vaccination immunity and, of course, behavioral changes. So many of the same conclusions that we have drawn here, just using an entirely different approach. All right. What about the future? And by the future, I mean the recent past, because our study ends in 2022, and I'm interested in Mpox in 2023, 2024, and 2025 and going forward. So what does the future of Mpox hold for us? Now, what I'm going to talk about now is not part of the manuscript. This is sort of some just additional investigations that I think help focus our thinking going forward. So, in 2024, when we started to see an uptick of cases in New York City, I went to the CDC's website to look at the national case data, and they had stopped reporting it as of February 27th, 2024. The good news is, well, they've started reporting it again now, and we can see that in 2024, you can see there mid-February, cases have been steady with about 10 a day across the US, maybe a little less. So basically, we've had steady rate of infection in 2024, which is substantially more than we saw in 2023. So we had about a 9-month lull in cases, and then the rates more than tripled compared to its lowest point. So we are definitely seeing sustained transmission of Mpox in the United States in 2024. But these are national numbers and these are very low. So the question is, how is Mpox persisting? And I don't have a great answer for you. But here we have our San Diego numbers, where you can see we had our main outbreak in mid 2022, and then essentially no cases throughout a good chunk of 2023 until we had a spike around October of that year, then another lull, and then another spike in the middle of 2024. So the question is, why is this happening? Where are these cases coming from? How is this virus how is it persisting? Well, the one thing I can tell you about San Diego is that all that big spike in 2023 was likely due to a single introduction. Now, you may remember in 2023, there was talk about a large spike in cases in Illinois, which you can see here, and reconstructed the history of this virus along a tree. So the light sort of pea green is Illinois here. The colors were automatically chosen, just the program. And then you can see the virus transitioning into red, which is Greater California, and then into purple, which is San Diego. So we seem to have this virus going from Illinois to California to San Diego. Well, we see a lot of cases and sampled genomes during that period in September and October and November of 2023, and then the cases died down again. So that didn't seem to be just a fluke, and that seemed to be the result of a single and sustained transmission cluster. We also see something very similar in New York City, where cases were very, very low throughout 2023. And then starting in October 2023 and January 2024, the rate about tripled. And what we found in New York City is that this tripling of the rate was associated with two very large transmission clusters. And you can see it here. Yeah, Nettie, that's interesting. New York City really didn't have a lot of cases in October of 2023. So there's just one New York City case there. It sort of was really in a lull, and it wasn't until afterwards that it picked up, mostly due to this cluster, which contained over half of all cases in New York City in the first quarter of 2024. And you can see here it's spreading Los Angeles County and Washington State. And then we also see sort of another sort of medium-sized cluster in New York City. So this virus is finding a way to continue to persist in clusters, and then seems to be spreading around the country after it takes off in one area. So it's not like we have low level of sustained transmission. Now, this is just my hypothesis right now. We still need to test this, is that we don't seem to have sustained level of transmission across the country, but we seem to have sort of bursts in one geographic area that tend to spill over to another, which lead to bursts there. And the virus is sort of jumping around from population to population without being sort of maintained in any single area of the country. So what we have is a sort of loosely connected sexual network across all of the United States. So I'm really glad CDC started reporting the daily data again. All right. So answer the big question here. Why did Mpox peak at the end of July 2022? Well, I argue it's the network, it's the network, the network. And the same factors that govern STI and the same lessons we've learned from over a decade of HIV, 2 decades of HIV molecular epidemiology are paying dividends when trying to understand novel emergent pathogens like Mpox. So with that, thank you for coming to High Rounds, and I'll take any questions you may have. Thank you.
Wait, this is your brain. This is your brain on drugs. Isn't that what that image is?
Any questions?
Just the any questions, right? Thank you. Sorry. Fabulous.
Yes, that's exactly what it is. It's
Fabulous.
nice.
Sorry, I've missed the any questions part of
No,
it.
that's all right. I present that at conferences, and people, especially if they didn't grow up in the U.S., they're like, "Why are you showing
Why are you talking
eggs?"
about drugs? Yeah. Okay. Sorry. That was incredible. As always. Which is lots of questions. Okay, so let me
Okay,
start
I
digging.
got the.
Do you,
Do you want to?
why
You
don't
can
I
do the
read them to
chat.
you so you can think?
Okay.
I think the first one came up. I'm just going all the way back to the top. I think the first one is from Nettie with the I might have expected more New York City represented in the Illinois tree. I'm not, I don't know
Hmm.
if something, but I'm not sure.
Yeah,
Yeah,
so
so
so no,
yeah.
yeah,
Oh,
you would think that, but so like as I mentioned, New York City had really, really low case rate during that time, and there was only one case that was in there. Now when we have a big New York City cluster, we are seeing Illinois come out of that, but I think it's just but these are like I'm just starting to get into these dynamics in the 2024, and the data are much more sparse now, not just the case numbers, but also the number of jurisdictions that are reporting genomes have gone down.
I know that our fearless chief just unmuted himself. Go ahead.
So, a few things. I'm just curious about the concept of the networks. Do you,
Mhm.
in the model, individuals in the model, are they just always in a specific network, or do they kind of move in and out of networks? Because what happened in New York, I mean, people would come in and there's like, oh yeah, I was in P-Town for, I don't know, Bear Week. And like, there was huge numbers of people that would report their exposure there, or
Mhm.
specific parties where lots of people got infected all at once. So, I mean, some of these networks were just like specific time, place events rather than sort of like an established, ongoing, connected
Yeah.
network.
Yes.
Does that matter
That
for the model?
Um, Not for this model. We're sort of assuming the model assumes a static network, but all of the and over a period of time, so over the course of your sort of infectious period. Um, so we're not allowing it to sort of rearrange, and given the sort of short duration here, we consider sort of like any sexual partner you had in that sort of month between infection and transmission. Those are your connections in the network, and with some probability of transmission. So we're not doing a dynamic sexual network. But as is evidenced by the lack of overlap between the HIV and Mpox network, uh, networks are dynamic, are very dynamic, especially over the long period of time. And I think that to incorporate more realism into a model that would be very helpful.
Yeah, I mean, I think in the Mpox trial, I mean, we've had a couple people that have been diagnosed with HIV at the same time, but it's been pretty rare. So it does seem that these are not like happening at the same time, like you suggested. And then I'm just wondering, is there a way to kind of model how vaccination, I totally agree that vaccination did not lead to the end of the epidemic because the same thing happened in multiple countries where there was no vaccination at all. And I'm just wondering, is there a way, knowing what the vaccine effectiveness is, how that kind of shaped these mini outbreaks going forward? So instead of having the huge outbreaks that we originally saw, I mean, perhaps the vaccination, if people in these highly connected people, if they tended to be vaccinated, how that would have impacted the epidemic.
Yeah, that is something that we are currently looking at for the resurgence in 2023 and 2024. We do see people with 1 or 2 doses of the vaccine in these new clusters. We don't really have an idea of the level of protection because if from the cluster analysis, because if you get infected and you've been vaccinated, then it's a breakthrough. But they may be less likely to transmit, and that's something that we're actively investigating with the health department.
Yeah.
And then what does sort of that baseline level of people who are vaccinated and don't end up in your cluster, because those are your success cases. It's the people who you don't see who got vaccinated, who never get infected. That's what we're really sort of looking for. And we haven't gone down that road yet.
And then just to say that in the clinical trial, we are getting, I think around 20% of cases are enrolling in the clinical trial, and 80% of those we have biospecimens on. So just thinking about what the opportunities are to take the sequence data over time from the clinical trial that's paired with some epidemiologic and clinical data, that's I think no one's really sort of claimed that analysis. So I think it is a really good opportunity.
Okay. All right. Thank
I'm going to
you.
start doing some I don't know if I can do it in real time anyway. I just threw something in so people knew what trial Dr. Wilkin was talking about. Let me go back up again. Did you see Susan's question? Is it possible to compare clusters with the same growth rate and compare the growth rate over time among clusters who received vaccine and those who did not?
Yes. I think that would be really interesting. Unfortunately, I mean, in New York City, those 2 clusters I showed you basically encompass like 80% of all cases with a sequence in New York. I mean, it was just 2 big clusters, but I think we can look at the parts of the cluster where people had vaccines and parts where they didn't, and see if you get less growth in the vaccinated part. I think, as you point out, the confounder is that the people who got 2 doses of the vaccine are the people who are sort of most acutely aware of their risk status. And that might counteract any protective effects sort of in the aggregate. But I really want to look at that. And I think we can also look at that in San Diego as well. But yeah, that's
big sample size.
Well, and fortunately, our numbers are still pretty small week to week, which is, that's good. I'd rather, not have the data than have too much of it.
Well, and then I think those are the next 2 questions, which I think you're answering. But, did vaccination help prevent Mpox resurgence past 2022? And would better vaccination eventually be able to abrogate the spillover of clusters and ongoing national, international transmission?
Yeah. So, I mean, I think one idea is that, with waning immunity from the smallpox vaccine, we're now, as a planet, more susceptible to Mpox. Now, the question is, we had Mpox show up here on prairie dogs in the early aughts, and we didn't get large outbreaks, but that may be basically the human component of densely connected sexual networks versus people that owned prairie dogs as pets. Not to say anything about people who own prairie dogs as pets. But that's not sort of the type of community that's necessarily going to lead to sort of these large outbreaks. And we could see declining immunity. Haven't seen great stuff on actually, if anyone knows about smallpox vaccine effects in Mpox. There was a lot of talk about it, but I haven't. People have good papers on that I've missed. Send them my way. And Abe asked about spillover, and absolutely. I mean, just to editorialize a bit, sitting on hundreds of thousands or millions of doses of a vaccine that could have been used in a population that has zoonotic pressure from this virus, and they just sat here in freezers, I think is inexcusable. And it's here, and we need to deal with it now, but we also need to focus on, clade 2 in West Africa, and also especially clade 1 in sort of central now Eastern Africa. So this really needs we need, I mean, this is a zoonotic pathogen, and we have a pretty good idea of where it's hiding. Jeff called it the biggest clusterfuck with bundled public health responses. I say, if we learn nothing from COVID, which we didn't, I really think that we could have gotten ahead of this. So I share your sentiments here. I think
Well, it seems like we have gotten ahead of it in the United States. I have to be careful about because obviously, I mean, it's a mess that what's happening internet, or at least in the DRC and other areas of Africa. But it does seem like, based on the numbers we're seeing, that they are small numbers coming in, that the response. I just want to be careful about how we're saying that.
Yeah, no, but I guess I'm thinking about this from prevention of sort of zoonotic pathogens and their spread, and we are not. This thing was probably spreading in Nigeria for years.
Yes.
Going back, probably 5 years before it made it into the MSM population in Europe and North America, and it's still spreading there. So that's my editorializing.
Yeah, and I don't know if Doug is still on the call. Doug, would you share what you told us yesterday at our talk about the vaccine availability in the low and middle income countries and where things are?
Right. So the vaccine from Denmark, the Junios vaccine for monkeypox, was approved, 3-4 years ago in Europe and North America. But the obvious place where its distribution would have the biggest effect on morbidity and mortality, as well as perhaps transmission of further epidemics, would be in the 2 centers that are spreading in Africa. And for reasons that only people who study bureaucracy know, the WHO has decided they need new or different or special data from Africa before they go through an approval process, as if it's not an emergency. Gavi, which is the organization that makes available and distributes all sorts of vaccines, from childhood diseases to everything else, throughout low and middle income countries, has got vaccine and is poised to do that. There are African investigators who are desperately understand and need to distribute these things, and even the Danish company, I think Nordisk, has said they've set aside 500,000 doses or something like that, but bureaucracy has obstructed public health, and the company said they will only scale up if there's an approval and mechanism to utilize the stuff they produce. So this is doing everything we can to permit. I mean, the Clade 1 really hasn't made a major impact yet, but it's more virulent than Clade 2, and it looks like we're almost inviting it to spread further rather than intervene.
Thanks, Doug. Joel, any last thoughts?
No, other than, we're still very, we're continuing this work. This is the first of hopefully sort of many investigations we're going to do on this. So, and I think with the public health emergency, a lot of local health departments here are very interested in doing more of this work. So, it will continue.
Thank you so much. And I just wanted to remind everyone we do not have high rounds next week. Please register for San Diego CFAR Research Day. It is on the high round schedule that Marvin sent out where you can get the link and register. So take a break from us, but enjoy research on campus. So thanks, everyone. Have a great weekend. Thank you so much, Joel. Bye-bye.